Hi, I'm Geoff Fudenberg, and I'm excited to present work with David Kelly and Katie Pollard on predicting 3D genome folding from DNA sequence. So genome folding poses a really interesting problem, both for the physical challenges it presents and the biological processes it controls. This is an electron microscopy image of a section of a mouse cell nucleus. Inside the nucleus, there are meters of DNA packed into a micron-sized object. My favorite analogy is this is about 50 miles of thread packed into a yoga ball. Emerging work now connects genome folding to processes that range from communication between enhancers and promoters, to digit and limb development, to systemic developmental diseases like Cornelia de Lange syndrome, and VDJ recombination and the generation of immune repertoires. Genome-wide chromosome conformation capture experiments, including Hi-C and Micro-C, have greatly advanced our understanding of how the key proteins CDCF and cohesin organize the genome. Hi-C experiments act as a sort of molecular microscope, and they convert pairwise physical contacts in 3D space into molecular products that can be read out using high-throughput DNA sequencing. So this animation shows Hi-C data starting at the level of multiple chromosomes and then zooms into the scale of a few million base pairs or megabases. The axes represent the genomic coordinates of some of the genes in the region indicated by their name and orientation. And the color scale for this Hi-C data represents the frequency of contact. So darker colors reflect more frequent contact, and lighter colors indicate less frequent contacts. At the megabase scale, where we are now, we can see that the genome is folded into a rich variety of local patterns. And this includes boundaries that separate adjacent domains. This is an example of a boundary, as well as peaks. Here are some peaks of high contact frequency between particular pairs of genomic loci. So CDCF is an 11 zinc finger protein that binds specific locations across the genome with an approximately 18-letter DNA motif. It's thought to halt this process of loop extrusion by cohesin in an orientation-specific fashion. And this interplay is what's thought to create those rich patterns of peaks and boundaries across the genome that depend in part on the layout and strength of the CDCF sites. Still, how to predict the consequences of perturbing the DNA sequence in any individual CDCF site or other regulatory element on genome folding remains an open challenge. And why? So first, not all boundaries have CDCF. Not all sites CDCF is at the Hi-C peaks or boundaries. And the height of these peaks is only kind of roughly correlated with the strengths of boundaries. So for example, these appear to be fairly robust peaks in the ChIP-seq data, but there doesn't appear to be much going on in the Hi-C map below it. So second, to solve this problem, we need to distill and propagate information over 6 orders of magnitude, from the scale of base pairs to megabases, where these patterns become evident. So here, to make this a little more visceral, I show the DNA sequence inside just one bin in the map, with positions of 2 CDCF motifs in blue and red, respectively. But what we really need to do is pick up hundreds of CDCF needles in a millions of base pair haystack and figure out which ones and which combinations of them actually matter. So what saves us from this DNA deluge? In the past few years, convolutional neural networks, or CNNs, have emerged as powerful tools for modeling genomic data. The general recipe involves taking input sequences, transforming them with many convolutional layers, and making predictions for a wide variety of genomic tracks and features. So CNNs now show state-of-the-art performance for RNA binding, transcription factor binding, DNA accessibility, and more. And importantly for us, CNNs can actually learn features from the data as opposed to needing them to be exhaustively designed by hand. And interestingly, some of the same architectural innovations that have been really fruitful for images and natural language processing are also seeming to really help performance in genomics as well, including things like dilated convolutions and residual connections between layers. So this is the 50,000-foot view of our approach. We take in a megabase of DNA sequence, one hot encoded as ones and zeros, transform this with many convolutional layers, and output a quantitative prediction for the contact frequency between any pair of regions in that region at 2048 base pair resolution. The network has a trunk, which pools input sequences to the 2048 base pair resolution, and then shares that information across a sequence with dilated residual convolutions, followed by a head that transforms from this land of sort of 1D profiles to the land of 2D maps. We trained this model by splitting the human genome up into thousands of non-overlapping megabase chunks with an 80-10-10 split for training, validation, and test sets. And just a few key things that allowed any of this to work were: one, taking advantage of the symmetry of contact maps; two, sharing information across the sequence, not just in the network trunk, but in the head as well; three, making really heavy use of data augmentation by shifting and reverse complementing our input sequences; and finally, squeezing the limits of what can be put into GPU memory, because the output of the first layer, a 1,000,000 by 48 matrix, is pretty large. So our poster covers this introduction and then details some of the exciting applications and results that we found using Akita. And this includes, first, extracting predictive base pair level information by in silico mutagenesis. So doing things like mutating the base pairs underlying individual motifs in the genome, predicting the impact of structural variants like deletions and inversions, and predicting genome folding across species. So we thought it was actually pretty cool and exciting that when we did this, when we took a human trained model and made predictions into a mouse genome, we found a lineage-specific impact of SINE V2 retro elements on genome folding. So in the future, we envision that approaches that build on Akita can advance our ability to design functional screens, model enhancer-promoter interactions, prioritize causal variants in association studies, and predict the impacts of rare de novo variants. We'd really be thrilled to hear any of your questions, comments, and suggestions. So please check out our poster, please check out our preprint, and thank you very much.